Hello everyone, welcome to OncoNerd, where complex cancer therapies are broken down into simpler bits of information. In this video, we will review another common chemotherapy drug, Fluorouracil. By the end of the presentation, you will have learned about these key points. We will start today with a very brief biochemistry dive to leeway into our talk about Fluorouracil. Our cells contain genetic material that code for the production, growth, behavior, and everything about a cell. That is called DNA. A copy of this genetic code is made, which is called RNA. RNA is then used to code proteins to be made, which go and do various functions for the cell. DNA and RNA are made up of chemical structures called nucleotides, which are various ring structures as pictured here. I would like to bring your attention to uracil, which is the second structure in the diagram under pyrimidines. There are 4 different nucleotides that are put together in various sequences to make up DNA. These are known as adenine, cytosine, guanine, and thymine. A copy of the DNA is made into RNA, a single-stranded structure, which are made up of 4 different nucleotides again, but this time adenine, cytosine, guanine, and uracil in place of thymine. Uracil is similar in structure to thymine, but without a methyl group. 5-fluorouracil, or 5-FU, is an analog to uracil. It contains a fluorine group in place of a hydrogen. 5-FU was developed by scientists in the 50s when they found that uracil concentrates in high amounts into tumor cells. By creating 5-FU, they are able to target this, preferably high uptake, into tumor cells. It was first FDA approved in the 60s and is now used in various chemotherapy regimens such as FOLFOX for GI cancers. 5-FU can cause RNA damage because its metabolite is used in place of a uracil metabolite structure. It also causes DNA damage. Uracil can be formed into thymidine, which, as a reminder, is used in DNA. And this process is done by thymidylate synthetase enzyme that methylates uracil into thymidine. 5-FU metabolite, specifically known as F-dump, inhibits this enzyme and blocks the creation of proper thymidine from uracil metabolite dump. 5-FU metabolite are also incorporated into DNA, which cause direct DNA damage. 5-FU is mostly degraded in the liver by DPD enzyme and excreted mostly through the respiratory system, but also through the kidneys and the biliary system. 5-FU is contraindicated in these conditions listed, such as poor bone marrow function and complete absence of DPD enzyme activity. Side effects include cardiotoxicity and ECG changes, bone marrow suppression, diarrhea, mucositis, and hand-foot syndrome. Those with DPD enzyme deficiency may experience these side effects sooner and more severe than those who do not have the deficiency. In cases of overdose or severe early onset toxicities, uridine triacetate is given as an antidote. 5-FU is administered as an IV bolus or continuously through an infuser pump, which is a portable pump that is worn, and it delivers the drug over days by the pressure of an internal balloon. It can also be given as topical preparations. It is dose adjusted in the elderly, those with DPD deficiency, and those with liver abnormalities. 5-FU may interact with various drugs such as other cardiotoxic drugs like anthracyclines, brividine, surovidine, and analogs, which inhibit DPD and reduce the breakdown and clearance of 5-FU, thiazides, metronidazole, allopurinol, phenytoin, and warfarin. Thus, checking for drug interactions is imperative. And that's the end of this video. Thank you so much for tuning in. Please give this video a like and subscribe. Feel free to comment below for any oncology drug topics you'd like to see being covered in this channel. Hope you have a wonderful day.